des lipides. Deuxièmement, il a des intérêts médicaux qui sont pour l'instant distincts des nôtres, mais qui peuvent les rejoindre. Et en particulier dans la réflexion sur le métabolisme des lipides, l'obésité et le microbiote intestinal. C'est quelque chose, petit à petit, le lien entre le microbiote intestinal et l'obésité est en train de se faire exploser. Je suis en train de faire un papier. Le premier papier qui soit sorti, en fait, on met les deux, microbiote et obésité. Le premier papier, c'est Leigh Gordon en novembre 2004. Et il y a 2 jours, on refait la même chose, et il y avait 330 papiers, dont la moitié depuis 1 an. Donc le domaine est en train d'exploser, et nous, les points qu'on explore, c'est antibiotiques et probiotiques, et obésité, et bien entendu, on est intéressé récemment aussi les lipides et les glucides avec la marque, et les lipides maintenant ont un rôle dans cette prise de poids associé à la manipulation du microbiote. Merci.
Merci pour votre invitation. Donc je vais essayer de faire le lien de ce qu'on fait au niveau médical ici à la Finlande avec les la gestion des lipides, le métabolisme des acides gras, et essayer d'aller petit à petit vers comment on peut intégrer le microbiote. Je vais à l'enlever. So the presentation today will be fat digestion in humans, and I'm showing you a few connections with obesity in humans, selected ones. So I will not tell you about genes involving obesity. This is not my specialty, and I don't think there is a gene for obesity or several genes for that. It's more complex than this. And today I will try to focus on a very simple process for explaining obesity in humans. And we have been working on this track during the last 20 years with the Department of Experimental Just before entering into the detailed presentation, I have a few slides to introduce the laboratory because it's on a CRS campus in the south of Marseille. It's an area where there is also microbiology, but this is a different way of doing microbiology. So it's also important, I think, to introduce what we're doing in this lab because there could be some other interest for you. So in our lab, we are working on lipolytic enzymes and lipids. This is a long-standing tradition in Marseille. We're working on these enzymes. It lasted for about 70 years. And actually, there was a co-development of the biochemistry of lipolytic enzymes. And at the same time, some medical doctors in Marseille, the gastroenterologists with the group of Professor Sarr, they have been interested in the physiology of the pancreas and related disease. And there was a tight connection with lipid metabolism, so the biochemists and gastroenterologists, they have been always working side by side. So in our lab, we are covering the entire field of lipolytic enzymes from molecular aspects. So we are doing structure-function relationships on some very peculiar enzymes. So that's the molecular level, with the structures, enzyme kinetics. And then we can also study the physiological role of these enzymes, and there are connections with several applications in medicine, chemistry, other fields. So we have, for this molecular abscess, we have a set of experimental approaches, and the particularity of the lab is to develop, always try to develop, every time there is a new enzyme, lipolytic enzyme identified, develop specific method for the assay of this enzyme. It can be an enzymatic assay, it can be an ELISA, and this has been very useful for them working on physiological levels, and particularly with medical doctors where we are doing human clinical studies, and we are collecting samples from the GI tract, so we can assay specifically enzymes from the stomach, from the pancreas, and maybe one day some enzyme from bacteria. Concerning application, I already mentioned there is application in medicine, so you will see one of these applications today. This is the use of lipase inhibitors for the treatment of obesity. But there are also biotechnology applications. There are lipases in the washing powder, so every day when you wash your clothes, you are using lipases. You can use this enzyme in environments, and there are also a lot of use of this enzyme in chemistry for making a specific reaction to find chemicals. So we're studying several lipases, but among them, there are more interesting enzymes. So we have the enzyme from the GI tract, pancreatic lipase, gastric lipase, enzymes that are related to these ones that have been discovered more recently. You have also hormone-sensitive lipase you find in the adipose tissue, which is very important for the mobilization of fat. And there is a connection there with other diseases like type 2 diabetes. And of course, microbial lipases. So they are well known for various applications. I mentioned the washing powders. Today there are many targets, for instance, in tuberculosis research, because we know Mycobacterium is producing a lot of these enzymes. We don't know so well today what are the physiological of the enzymes, but fat mobilization seems to be important for the dormancy of the enzyme and then the reactivation, the bacterial reactivation. So all these enzymes, they are connected with important physiological functions and the pathologies that are related to these functions. So you have lipolysis in adipose tissue, and if you generate too much fatty acids, you can have insulin resistance. That's a factor in type 2 diabetes. So if you can inhibit the lipolytic enzymes involved in the release of fat, you can try to develop drugs against type 2 diabetes. Lipolysis in the GI tract, so that will be the topic of today. If you block the digestion of fat with lipase inhibitor, you're going to reduce the uptake of calories, and this is something equivalent to low-calorie diet, for instance. On the contrary, people who do not digest very well fat, and these are patients with pancreatic exocrine insufficiency, and this could be chronic pancreatitis, cystic fibrosis, and in that case, instead of using inhibitor, we are developing enzymes for replacing the enzymes from the pancreas that are missing. And so here there is also an interest for microbial enzymes because they are more easy to produce at a large scale, at industrial level. Some of them, they can be isolated from sources where they're highly resistant to drastic conditions, such as low pH, and they could be good targets for developing enzymes for use in humans. And I already mentioned that there is an interest for lipolysis in microorganisms, but also with microorganisms. And already lipolytic microorganisms can be used in various biotechnological processes. So I will go quickly on this one because it summarizes the various applications. So at the lab we have 4 research groups. Mine, which is more dedicated to the molecular mechanism of enzymatic lipolysis and interaction with lipids. The group of clinical studies of gastrointestinal lipolysis, the group of Stéphane Canard, that you have seen there, I think last year, who's working on lipolysis and bacterial pathogenicity. So he has subjects that are closer to the interest here, and he's the one working on mycobacterial lipolysis and how these bacteria interact with lipids. And we have also a cooperative library with a small biotech company in Marseille called Germ that is using microorganisms and lipolysis for various kinds of applications. Here we have in more detail the group of chronology, which also involve Professor Jacques Salle for the Children's Hospital. And so since 20 years we are welcoming their students, master degrees, some of them also PhDs. Recently we had several of them who work on premature babies, and we study fat digestion in premature babies. And more recently Camille Jacino who work on the methods for quantifying lipid in feces, and particularly the release of fatty acids. So there we have also some connection with the microbiology. So now let's enter into the science for today. I would like to focus on only 3 points that are the following. Too much fat in foods. So it seems obvious, but I will show you how it's connected with the digestion of fat and how you can digest as much fat as you eat, which is already a mechanism. Then another point that is less known, that is currently highly investigated, that's the kinetics of digestion, and particularly the kinetics of fat digestion. And here I know it's too fast fat digestion, and I will explain why. And then I would like to conclude on the role of fatty acids, which are often considered as vehicles of calories. In our diet, the fat contains more calories per gram than carbohydrates or proteins. So they are major vehicles for calories. And their impact on obesity is what usually considered as being connected to this property of fatty acid, is that they contain a lot of energy. But that's not all. In fact, these molecules, they can be also signaling molecules. There is a high diversity in the structure of these molecules. I will show you one example to illustrate that. And so we know that they can have some fine-tuning of metabolism. So concerning the first point, I will show you some results concerning the inhibition of fat absorption in humans. So we have been able to do clinical trials with test meal in humans, and we study various kinds of meals, and we have been using also the lipase inhibitor, or LISA, which is used today as a drug for the treatment of obesity. This is actually the main anti-obesity drug on the market. You probably heard about it. I will show you some data on the kinetic of fat digestion. And in fact, our interest for that came from the study we performed on lipase inhibitor, where we had some experiments very difficult to explain. And this was based on the kinetics of lipolysis versus kinetics of inhibition. And if you digest fat too fast, then there are some satiety mechanisms that are not actioned in the GI tract. And this is a mechanism that is linked to the food, the structure of food. So I will show you today how the structure of fat in food is very important. And the idea is that you have to think about all the manufactured food we are eating today that's changing drastically the way the fat is presented to our organisms. And this has a major impact on the obesity, rather than genes, etc. Here I will again show a few examples on how fatty acids they can have a fine-tuning on lipid metabolisms or in metabolisms in general. That's not only for lipid metabolism. They can regulate, for instance, gene expression. So if you are not in the field of lipids, sometimes this property is not well known. And I will show you an example where some specific fatty acids they can control the storage of fat and its deposition in various tissues and organs. And if you change for an isomer of this fatty acid, you can completely change the localization of the fat in the body. And that's the particular property that's linked to the structure of fatty acid. So that's 2 points today: the structure of food, the structure of fatty acids. And I will finish with some reflection on how we can include the GI tract microbiota in all these processes. So if we consider the digestion of fat in humans, so as I told you, we eat too much fat. Fat is about 100–120 grams of fat per day in Western countries. So it represents about already half of the calories we need every day. And this starts in the stomach's reaction of gastric lipase, which is the first enzyme on the forefront. And then when the diet mixed with gastric section is emptied into the small intestine, we have the action of pancreatic enzymes. The major one is pancreatic lipase, and lipase is cofactor, so this has been known for a long time. And you have also some other enzymes with pancreatic secretion, like PLRP2, carboxylasparagolase, that have a minor importance in this process, and also different specificities. But when I will start showing results on lipase inhibitors, you have to know that all these enzymes, they are inhibited by the same inhibitor. They have a similarity in the active site of action. So these enzymes, they are involved in gastrointestinal lipolysis, and they will generate the lipolysis products that are absorbed at the intestinal level. So this is the reaction of enzymatic lipolysis. This starts from the triglycerides you find in our diet. So the triglycerides represent 99% of the fat we have in diet. Then you have minor lipids like phospholipids, galactolipids, etc. From the point of view of the diet, they are not so important in providing the fatty acids. So today I will only focus on the lipase and the triacid acid. So the lipase, they can release one acid chain from the triglyceride and give rise to diglyceride. And the same enzymes, they can go on to release a second fatty acid, and they give monoglyceride. And at this stage, you have already the product that can be absorbed at the intestinal level, that can be transformed in micelles with bisols, and transport towards the intestinal barrier, where they can cross it. And these are the free fatty acids and the monoglyceride. For the diglyceride and triglyceride, this is not possible. They cannot be absorbed like this. Some lipases, they can also release the third acid chain, and you can give rise to glycerol. So normally we think it's not essential for our metabolism, since we can absorb monoglyceride, but we know that some enzymes, they can do it, and you have a fraction of triglyceride that is totally at the rise. And when you analyze the lipid in the blood after reabsorption, you can see that there is a mix of fatty acids. One important point here, when you're studying lipolytic enzyme, I will not enter too much in the structure-function relationships today, but there are a few notions that you have to know. So we start from products that are insoluble in water. So the enzymes, they have to go at the oil-water interface. So they have a peculiar interaction with the substrate. And as soon as the reaction goes on, you transform the fat into products that interact with water. They can fade, form micelles, lipid crystals. And that's the reason why they can be mixed with the biolipids and then transported into the GI tract. That's essential for the mechanism of absorption. So the idea is to show you how the structure, the physical chemistry of fat is important for the action of enzymes. And you will see later on that this has also an effect on the action of the lipase. So the lipolytic enzymes and the lipase in particular, they are soluble enzymes, highly soluble in water, but they are able to change their structure to bind at the oil-water interface, where they carry out catalysis. And among all lipolytic enzymes we know, usually the specificity for a given substrate is due to this step here of recognition of the lipid structure, rather than a specific architecture at the active site level of the molecule. So it's very important to know the physical chemistry. So you have all these steps. And still in connection with the structure of fats, if you think about how the fat enter in our GI tract, usually if you aspirate the gastric content, you will see large droplets of fat as soon as the food starts to be digested. It's because in the meat, if you use cooking oil, or even if you have butter that is an emulsion, but then if you melt it, you will start with droplets. You have usually large droplets. And when you have large droplets, you have a low accessible surface for the enzyme, and you have a low enzyme turnover. This is known by enzymologists working in this field since a long time. This was already theorized in vitro, but then we have seen it in vivo. Then you have the process of emulsification. So if you read textbook about lipid digestion, you will always see that when the fat enters into the GI tract, there is an emulsification process, mainly bisols, dietary proteins, phospholipids, that give rise to the emulsification, so the dispersion of the oil phase in the water. This generates high specific surface, so you have more surface per unit volume for the absorption of the enzyme, and the result is that you have a higher enzyme turnover. It's like when you make a Michaelis-Menten curve and increase the concentration of substrate. That's the same phenomenon when you increase the surface for lipases. And I think that's one of the principles in this area, and this is sufficient for understanding the talk of today. And so the emulsification, this is a process that can take place in the GI tract with the bile. But you can also have a pre-emulsification in the food. This can be a natural pre-emulsification. Milk, for instance, it's good substrate for enzymes. But if you look at the food that are manufactured today, you will see that they contain a large amount of emulsifier, and among them you find lecithins, twins, monoglycerides. And so next time you go to the supermarket and you buy food, have a look at all the ingredients you have written on it, and you will see that they are used a lot. And even in foods where you don't think they are, And that's a major problem, I think, with nutrition today, is that if you don't cook yourself and start by this, you will face immediately these two, and this has a major impact on the kinetics of digestion and obesity. So now to show you more how it goes in vivo, I will show you some data from clinical studies we performed with René Roger. So this is our test tube. We have human volunteers. We work with healthy volunteers, also patients with chronic pancreatitis. And we are doing experiments with a test meal in which we know very well the fatty acid and the fat composition before doing the experiment. So we know how much we have, and we know the quality of the lipids. And so the principle is that we do the test meal with a gastric tube. This tube allows also to take samples in the stomach during the digestion, so you can have access to lipase secretion in the stomach, so lipolysis product. You have a second tube that goes in the small intestine, and we have an occluding balloon that we can inflate at the angle of trace. And upstream of the balloon, we have aspiration lumens, where we can collect all the sheen that's coming into the small intestine. And with the second tube, we can have access not only on the concentration, but also the output of digestive enzyme, the output of lipolysis product. We have also a marker in the meal that allows us to make some correction, because this is physiology. The recovery is not always 100%, and we need ways to correct the data when we are missing some of the material we introduce initially. But at the end of the day, we can make a vast balance of what we gain, what we recover, how much enzyme we produce, so we can have a full quantitative analysis of fat digestion in humans. Unfortunately, we can also have it in this way in the upper part of the small intestine. If you try to have a tube that goes lower in the small intestine, then already it's not pleasant for the volunteers. And the recovery also is very bad, so you cannot have access to the same quantitative tools. So in that case, the best is to go at the end of the tube to see how much lipids you have in the feces, if the fat is not digested, and you can also analyze it. In animal, of course, there are some other studies where you can look at various stages of the GI tract. So when you place the tube, you have an iron guide in the tube, so you can see it. And here I put some red and blue dots to show the tube going into the stomach, so you see the grid curvature of the stomach. And you have the aspiration lumen that is here at the base of the stomach before the pylorus. And you have the duodenal tube that crosses the stomach, goes through the pylorus, enter into the duodenum. So you have the ampulla water, which is not far from there, with the arrival of pancreatic secretion and back. And the tube goes up behind the stomach, where we have the junction between the duodenum and the jejunum. And that's where we inflate the balloon. So from the samples we can collect during digestion of a meal, here you see extraction of gastric and duodenal samples that you can easily recognize by the color due to the bag. We can do various assays. So for the lipase, as I already said, we have specific lipase assays where we can measure the activity of gastric lipase independently from pancreatic lipase. And we have also ELISA with antibodies we develop with Immunotech. So for the same enzyme, we can have the total mass of enzyme that is produced, and we can have the activity. And this allows us to see the specific activity in vivo. And if you have, for instance, some degradation, inactivation of the enzyme, if you are using lipase inhibitor, for instance, you can see it by these means. So this is what I said. So at the end, you can have access to the lipase secretory outputs. And you can also have access to lipase inhibition levels if you use a lipase inhibitor during these experiments. Besides the analysis of the enzymes, we look at the lipids, and that's where we will see the work of these enzymes on the lipids we have in our diet. And so we have several techniques, ifr48 for separating triglyceride and lipolysis product, and the gas chromatography for watching in more details the type of fatty acids we have. So this type of chromatogram we have validated methods, and from that we can have access to the output of lipolysis products. So we can say how much fatty acids, for instance, we produce in the stomach, in the duodenum. So we can estimate the lipolysis level because we know how much fat we gain. And we can also calculate lipase-specific activities in vivo, and this is very informative. As I told you, I'm a chemist. I started working with test tubes and well-defined conditions for studying enzymes defined by enzymologists, and I can tell you the rate we measure in vivo has nothing to do to the rate we measure in vitro. It's 3 orders of magnitude lower. That is sufficient to digest fat. What we measure in vitro in the lab, if the rate of that digestion was the same, we could digest all our diet in 50 seconds. So it's very informative for an enzymologist to do this kind of experiments, because then when you go back to the lab and you are with your test tube, you think in a different way. So during the last 20 years, I think we performed about 150 experiments of the time. And so we have a global view of that we can include in the textbook on fat digestion in humans. So concerning the human gastric lipase secretion, we produce in between 10 to 25 milligrams of enzyme during the diet. So here our experiments last 4 hours in order that the stomach is totally empty. And we estimated that the lipolysis inside the stomach, if you measure all the fatty acids coming out from the pancreas here, it represents in between 10 and 25%. So you can see difference, and I will show you what are the conditions for this difference. If you think about the duodenal stage, we can have access also to the secretion of human pancreatic lipase. Here you have huge variations. So it ranges from 80 to 400 milligrams of enzyme per minute. So 400 milligrams, and the pancreatic lipase represents only 3% of the proteins in the pancreatic secretion. So you imagine the capacity of biosynthesis of the pancreas for producing digestive enzymes. That's grams of enzymes that cycle every meal in our guts. And that's also a point that I think among the things we have done that I think are important during the last years. When I started in this area, I could only read from articles in gastroenterology or other journals units of enzyme. And very often you don't have the conversion between units and milligrams. And from one lab to another, the method for assembling enzyme is different, so there is no connection between the units. That was a big mess for me when I started to do that. And so that was very important that we estimate how much in terms of mass we have. And then everybody agrees on this value. What we have seen is that the gastric lipase can still work in the small intestine. So there were some controversies about that, so we found it was there and it was active. It works in synergy with pancreatic lipase. That's more difficult to study because you can have that activity on the same enzyme and the same protein that are raised. And we measure the lipolysis level. So again, we integrated the flux of fatty acids and the angular traits here. And we have also variations between 30 and 60% of lipolysis. So you see that in some cases, the digestion of fat is far from being finished at the angular traits. In some textbooks, you will read that it's finished. It's because it's based on some experiments where people infuse emulsions in the small intestine, not in the same conditions. You will see that if we use emulsion, we go closer to this value there. But if you have a normal meal, we are closer to 30%. And that's why I think we need a small intestine of several meters long to complete the digestion. And we can see in some pathology with a small, short small intestine. It's right. So I will go quickly on that one. So the figure we have today is that if you take 2 triglycerides molecules in the diet, you need to release 4 fatty acids to generate 2 monoglycerides. Gastric lipase is responsible for 1, and pancreatic lipase for 3. So I think this is something we can remember. Then we can enter in more detail and why we observe these huge differences on pancreatic enzyme outputs. Now we are summarizing in this diagram here some big values obtained from our studies and also a few other studies performed in Switzerland. And we tried to see if there was a correlation between the output of pancreatic lipase in terms of milligram and the amount of fat in the diet. And you see the more fat you eat, the more enzyme you produce. It seems that there is a plateau here, that you already have a huge production. And you see the 80 milligram is there, 400 milligram is there. So these are the experiments we perform. If you take a meal in which you have 15 grams of fat, you produce around 200 milligram of lipase. If you have 30 grams of fat, you produce 400 milligram of lipase. So you have an immediate adaptation of pancreatic secretion to the diet. So it was known already that if you take rats, for instance, you give them a diet enriched in fat, when you watch the level of the messenger for the digestive enzyme, they will increase with the amount of fat. But this has never been measured really in humans. And here you have directed demonstration at the protein level. So this is like a slogan you find in many American restaurants. Eat as much as you can, and you will be still able to digest it. So how this works? So now there are various reports in the literature, and I think it's something well known now, that when you hydrolyze fat, you generate fatty acids. And fatty acids, they are secret analogs for one important gastrointestinal hormone, which is CCK. And so this has been lost. The main effector is oleic acid, actually. So through lipolysis, you generate free fatty acids. You trigger the CCK levels. You take a plasma sample and see that it's increased. And you will increase the pancreatic secretion. That's the information from this hormone. And so you have a group here where you can increase the amount of lipase. Among the experiments that have been made to prove this pathway are experiments performed with lipase inhibitors. So if you use a lipase inhibitor, you block the lipase action, you will reduce the level of the fatty acids. So you can expect to reduce CCK. And this is actually what was observed in the experiment with orlistat. You have a lower CCK level in the blood. And if you measure the output of pancreatic enzyme, you have a reduced pancreatic secretion. And these are our results we obtained at Marseille. We have similar results for other groups. That's the two types of meal we have been using. I will enter more details in this meal later. If you take a good meal, which is a complete meal with the fat that is homogenized and solid meal is made with hamburger, fries, beans, etc. You see the blue bar here, that's the controls, no inhibitor. In this set of, in this clinical study, we had about 250 and 200 milligrams of lipase produced. There are some differences in meals. And if you give the orlistat inhibitor, either the pure molecule dissolved in the meal, or the xenical-pellet, this is the drug you have on the market, you see that you have a reduce on the output of the lipase. So this is an indirect effect of the drug that was not envisioned initially. The drug was developed for blocking this step here and reducing the uptake of calorie. But the other indirect effect is that you reduce the output of the digestive enzyme, which in a sense it's rather interesting. It may be more interesting than this step here. So let's come now to the difference between the meals and how this meal finally have been very interesting for understanding several mechanisms. So usually in Marseille we're using a liquid test meal. The reason for that, you have seen you are using tubings. If you want to aspirate the meal from the stomach and the small intestine and you want to have a good recovery, it's better to have something that is liquid. So we have been using that for most of our studies, and still we start to work with Hoffmann La Roche on the development of the lipase inhibitor. And they said, okay, your meal is very nice, but we did all our clinical phase 2 study with American type meals. So we want an hamburger, french fries, beans, butter. So we had to prepare this meal. So of course, the patients, they couldn't eat, so we had to grind it to make small particles. It was not homogenized. It was given with a lot of water so that it could go through the tubing. And you will see that that's already makes a difference between this and this. So, if you take what we call the solid meal, or the normal meal, we measure that in the stomach, we will, the result of lipolysis will be about 9%. At the angle of trait, we will reach 27%. That's close to the 30% you have seen before. So we are far from a complete digestion of fat. In the titanium and helium, we have no access, that's a black box for us. But usually we know, and also from animal studies, that normally before we enter into the colon, we should have digested almost all the fat. The main reason for that is that the absorption will occur in the small intestine. And this is confirmed by the fact that usually in healthy people, the percent of ingested fat you find in the feces is usually lower than 4 expressed in percent of the fat you get. Then you can have expression of steatola in other terms, but this is what we use here. If you take the liquid meal, so you see the gastric lipolysis is much higher, 24%. At the angle of traits, we are already 60% of lipolysis. So we double the level of lipolysis at this stage. And still under these conditions, people are not known to have steatoria, large excess of fat in the feces. So this shows that pre-emulsified fat is digested more rapidly. So in this liquid meal, you have fat, but you have also a lot of lecithins and monoglyceride that are used for making the emulsions. And the reasons why they are used in manufacturing foods is that they allow to keep the emulsion stable on the shelf and in the market. Because very often the emulsion is like when you make vinaigrette or things like that at home. If you let it in the bench, you will have a phase separation. So in industrial product, you don't want to have this phase separation, so you put a lot of emulsifier. Recently, so this is a work actually that is a revision for publication in American Journal of Clinical Nutrition from colleagues in Lyon. They made the following observation, is that from our side we work only on the luminal part. But we have the idea that if we digest the emulsion faster, the rate of absorption can be also faster, and the appearance of the lipid in the blood, the chylomicron, should be also faster. And this is actually what I have shown recently. They have used in the human study dairy fat, and they gave 40 grams of this fat, either spread on the bread or pre-emulsified with lecithins. And you see the results they obtained in healthy volunteers, but also in obese subjects. Here, this is the concentration of chylomicron TAG in the plasma. So after fat digestion and absorption, you resynthesize the triglyceride, and they are excreted in the lymph and the blood in the form of lipoproteins. And this is what we are looking at here. And you see that with the dilution, there is a peak which is higher, and which is also faster than with the fat spread on the bread. And you find this also in the obese people. It's a bit higher. And also this people here is not shown because the zero already takes into account the difference with basal concentration. But they have already a basal concentration on TAG that is higher than in healthy people. So it seems that what we have seen at the digestion level is well connected with the absorption level, and the fact that the fat will go more rapidly into the circulation. So what is the impact of these kinetics? There is a mechanism of satiety that is rather well known and currently well investigated, is that when you ingest food, particularly fats, there are satiety mechanisms that occur when undigested foods or some lipolysis products that are not absorbed already arrive in the distal part of the small intestine. And this will trigger not only that one, but these mechanisms, they are rather well known, the production of gastrointestinal hormones that will have several effects on the motility of the stomach, the small intestine, so on the gastrointestinal transit. On the secretion of enzymes, so you will reduce the production of enzyme, and you have also a cephalic phase and you reduce food intake. So this is a mechanism that occurs normally during digestion, and you think immediately that if these products They are all made here and absorbed rapidly in the upper part of the small intestine, which has the highest capacity actually. You will not have this mechanism here. So, actually, I'm really thinking that this could be a major reason for obesity problems. This has been studied also on the long term. And these mechanisms of satiety, they occur on single shot during the meal, but there is also some adaptation. So if you eat usually and commonly meals that are digested very fast, you will not have these mechanisms. So the idea here is to, if you change the structure of the food you eat, you can have drastic change in the metabolism, in its regulation. So in connection with this, I would like to show the use of lipase inhibitor as drugs for obesity treatments. So the principle is the following. If you block the digestive lipases, you will block the formation of the lipases products. And so at the end, you will block the intestinal absorption. That's the reaction I've shown initially. And the result will be that you can have a reduction of calorie uptake. And also, even if you don't suppress totally the digestion, that you have some undigested product that arrive in the distal part of the small intestine, you can have an induction of these satiety mechanisms. So today there is only one lipase inhibitor on the market. There are many projects in the pharma industry for developing new ones. The one that is on the market is this one. It has several names. It's called tetrahydrostatin by the chemist. It's called orlistat as the active principle. And you have the two drugs that have a trade name, I will show you. So in fact, this is a beta-lactam with hydrophobic part. And this is a molecule that overall has properties close to diglyceride. That's probably why the lipase can get it easily. And you have a beta-lactam group here that reacts with the catalytic site of the enzyme and blocks the reaction. And actually, this is also a connection with microbiology. This molecule was isolated from Streptomyces pseudomonas. So it was in a stream of antibiotics, and actually, this work, the isolation was published in the Journal of Antibiotics. We don't know what is the function of this beta-lactam in the microorganisms. There is an interesting part, maybe you know it. But people are usually focusing on the application of this lipase inhibitor. They are not searching very well what is the function of these molecules in the microorganisms. And in our lab, we think that they are probably already antibiotics. And the target is microbial lipase. So the natural molecule can be transformed into a more stable molecule by hydrogenation. So the natural molecule is lymphatin, and you get tetrahydrostatin. And so it was first launched on the market in 1998 in the EU, and 1 year later in the US. So you might know this molecule by the two trade names. So the first one on the market was the Xenicor, introduced by Roche, who did all the discovery and all the developments. So this is a prescription drug, and it's still the case today. And 2 years ago, you have seen a lot of publicity for the new camera that was called Eli. This is marketed by GSK, but this is the same molecule. The formulation is different, the dosage is a bit different, but it has overall the same effect. These lipase inhibitors, today they are the main, so here it's mentioned pancreatic lipase inhibitors, but they also inhibit gastric lipase. They are the main, the top anti-obesity molecule on the market, and they have a part that is much higher than the other agents, pharmaceutical agent acting on the central nervous system. Most of them actually withdraw from use. So we perform similar experiments with the same meals, the solid meal, the liquid meal, with the Xenicor. And we obtain the following results. If you take the solid meal, you see that there is a very good effect on, so these are single shot experiments on one diet. This does not tell you how it goes in these people when they are treated in the long term. But on a single shot, we can have very impressive effect. So with the, we can have You see here we block more than 80% of gastric lipolysis, even if you know that in the stomach mixing is not huge, so the probability for having good inhibition is not high. And you have also a good inhibition of overall lipolysis and block traits. And so with the same diets, we had also a cohort of patients who did not have the tubes that we measured the steatoria after they completely digested the food. And we could measure that they were excreting about 40% of the ingested fat in their FSS. So this is what is published for Xenical on average, between 30 and 40%. Actually, if you put the drug in butter or in chocolate, you can reach 80%. But if you use that, you will be in trouble, because what is the problem today, that's the side effect of Xenical with medication and things like that. So that's probably why they never developed really the second generation formulation. So, and if you look at the results with the lipemia, that's where we were in trouble when we showed these results to Roche. You see at the gastric level, we have 8%. That has to be compared to the 24. So still we have a good effect on gastric lipolysis. So whatever the new gastric lipolysis is a good target. But if you look at the lipolysis at the angle of traits, here you have 48 instead of 59. And so that you have around 80% of the control. So almost no effect on these conditions. The same amount of inhibitor, almost the same output of enzymes. What is different, that's the rate of lipolysis. And you see, when you look at the steatoria, there is no steatoria, that's 4%. So that's what we have in LTP who are not treated. So we had in mind already that this could explain why in some cases people don't see any effect of this drug. And this was due to the type of thing. So it was very difficult to understand these experiments for the clinical trial. But for sure there was an effect on the pre-emulsification of fat in impairing the reaction of hypertension. So what we have done, since we collected many information on the physiology of fat digestion, enzyme concentration, pH value, we can reproduce today digestion in vitro, but this time under physiological conditions. We have the enzymes that we can produce as pure enzymes in the lab. We can also use human gastric juice and pancreatic juice. And so we have a very simple model that we use a lot now for screening inhibitors or testing various foods. We have a first phase where we incubate the meal with gastric lipase. We extract the lipid and we see what is the result of the action of the enzyme. And then after half an hour, we add pancreatic juice, bile. We change the pH to be pH compatible with intestinal functions. And we wait for 1 more hour. And usually that's the type of curve we will record for lipolysis level. So we do the same thing as the new humans, and the same analysis. We can analyze the residual triglyceride and the lipolysis product. And at the end, we can show the following plots. So if you take the lipid you start the gastric phase. You have gastric lipase. You can see that the enzyme remains stable. And under these conditions, we have a low at significant level we can measure of about 10–15% of lipolysis. That's our control without inhibitor. After 30 minutes, we add the pancreatic lipase, submit here. We see that as soon as we have pancreatic lipase, there is a jump in the lipolysis level. So that's because we have more pancreatic lipase. It has also a higher turnover. You have seen the ratio between gastric and pancreatic lipase, 10 to 25 milligrams during the meal for gastric lipase, between 80 and 400 milligrams for pancreatic lipase. So we have similar ratios here. So as soon as you put the pancreatic, you have gastric increase, and you reach almost complete lipolysis after a few minutes. If you do experiments with Xenical, so you mix the Xenical with the meal initially, you can block rapidly the gastric lipase activity. At the same time, you see that there is no more lipolysis here. When you add the pancreatic enzyme, you see also a decrease in the enzyme activity. And what you see is that you reduce the level of lipolysis, but this is not efficient and immediate. Initially, here you have a drastic increase, and then it slows down. that we have about 60 to 70% of the efficacy we can have without the inhibitor. If you look at the solid meal, we do the same experiment. We have gastric lipase, like this here, pancreatic lipase. You see here the rate of lipolysis is slower. So we come back to the initial information about lipolysis. The more the fat is emulsified, the higher the activity. The uniformity of the lipase is more active in the emulsified meal than on the normal meal. If you add the xenicol, same amounts, you block gastric lipolysis. Gastric lipolysis is blocked, so there is no effect on the meal of gastric lipolysis. This is a very big target for the inhibitor, and whatever physical chemistry you can eliminate. And actually, it's very different from the conclusion we draw before doing the experiments, like in vitro assays made in a different way. If you look at the pancreatic phase, you have a decrease in the activity of pancreatic lipase. Here, it's very similar. The half-time for inactivation of the enzyme is the same. So again, no effect on the food, on the rate of lipase inhibition by the inhibitor. But if you look at the activity here, there is no more activity. You can block almost 100% of the lipase inhibitors. So the conclusion there is that you have the illustration in vitro of the competition between the lipolysis process, the substrate entering into the active site of the enzyme, and the initiation process, the same active site being targeted by the inhibitor. So this inhibitor, they are named competitive inhibitor, and here you have really the demonstration of that. Sometimes when you follow courses of enzymology, it's not so evident to understand what it is, but here you have the demonstration. So the two processes, they go at the same time, they target the same active site. Here there is a preference for the lipid, and you have time to analyze a significant amount of data before the enzyme is totally inactivated. So you see that with time, the enzyme takes the inhibitor, and at the end it's completely abolished. But before being totally inactivated, you can have some activity. Here, since the rate of lipolysis is slow, you have time to inhibit the enzyme before the lipolysis is sufficient. So this is what I wrote there. So you see how the physical chemistry of fat and the way we prepare it can influence not only the rate of digestion, have an impact on satiety mechanisms, but also on the effect of the treatment. So I will try to accelerate to conclude rapidly. So the last point I wanted to mention to make the junction with the microbiome is that the fact that fatty acids, they are not only vehicles for calories. So you have several functions for fatty acids that are well identified. Of course, they have a nutritional role, source of calories, or storage of calories, essential fatty acids. They have also a structural role that is well known. They are part of the lipids-making membranes. Depending on the composition, you can change the fluidity of the membranes, control of receptors, etc. This is well known. What is also well known is that fatty acids, they are producers of important lipid mediators, like arachidonic acid, for instance, which is a precursor for prostaglandins, omega-3, which are precursor of resolving protectin, and here you have the molecules that are either a pro or an anti-inflammatory processes. And so they are also important for various diseases, but also for obesity. You have now more and more publications and studies on the fact that fatty acids, they regulate gene expression. And we know that fatty acids, they can be transported towards the nucleus, they can interact with specific nuclear receptor, and they have direct interaction with gene expression, very often with gene expression in the field of lipid metabolism. And probably because of that, you can see that you can find a specificity for various fatty acids, and this has an impact on fat deposition in various tissues. And when you change a little bit the structure of the fatty acid, you can change the fat deposition. So that's the principle of regulation of genes by fatty acids. I wanted to show you just one example, which is concerning conjugated linolenic acid. Again, this is not so well known, but next time you go at the pharmacy or the drugstore, let's search for these molecules. In France, very often you will not see CLA written like that. You have to look at the first bench when you enter the pharmacy where you have all this product to lose weight. And you will see that some of them they contain CLA. And because CLA, they have been described as fatty acids that decrease the mass of adipose tissue in the body. So these CLA, in nature, they are very rare. You can find them in milk and dairy products. Here you have rubenic acid with the cis and the trans double bond. Already trans, that's very rare. Normally we do not have trans ester bonds, double bond in the fatty acids. And you know probably that the trans fatty acids, they are called margarines, and they are connected with cardiovascular disease. But trans is not very good for health. So, but this you can find it in cow milk, and from what I know, it's from the microbial region. That's the bacterium from the rumen that produces this fatty acid you will find in the milk. And when you look at this chain here, you can have cis trans configuration for the double bond. You can see that you can have several positions for them. So you can imagine immediately the diversity of structures you can have with the fatty acid. That's huge amounts. And these are specific structures, like for other isomers where you have vertical antipodes. You can have the same features here. And so this means that this can be linked to a specific recognition in biological systems. So this one, for instance, is a non-natural isomer, that if you produce CLA by chemical means, and you do not extract it from milk, you will have a mixture of product. The same product as in pharmacy when you work on optically active compound, you can have a mixture. And so I will show you what has been done in the mice. So the reason why CLA are marketed today, I told you, is because they can reduce the mass of monotype. Here, this is study performed in the mice, and they look at the peripheral actually, I don't know why they look specifically at that part. But here you have the control. You have the mice that has been fed with a diet enriched in linolenic acid, that you find in many vegetable oils. You have the rubenic acid here, and you have this undesired isomer that you find actually in the marketed product. And in fact, this is this one that leads to a reduction of the mass of the adipose tissue. It's not this one. Whereas companies, they always claim this is the fatty acid that is important. So you can stop there, but there was a group in Dijon that decided to go further and analyze other organs of the mice. And the result is this one. If you look at the liver, with control linolenic acid and CLA, almost no difference. If you take this one, you have a huge increase of the size of the liver. It's full of fat, and for the medical daughters, it's very similar to steatosis. So it looks really like a disease. And also, if you look at the insulin, for instance, there is a huge increase in the insulin in the same animal. So it's a bit provocative, but it was to show you what can happen if you manipulate too much fatty acids, and this is what happens today in foods industry. You put fatty acids everywhere, even with omega-3, when there are controversies on their benefits for health or not. I'm sure that there are some areas where there can be some benefit, but you have to be very careful with these fatty acids. The structure is very important. It's not just fat. And so the last example I wanted to show you to conclude and make the connections microbiota. Actually, this is from a presentation I've seen last week on the production of CLA, so the same fatty acids like the lactic acid bacteria. And the pathway for this production has been well described in Lactobacillus plantarum. And the genes involved in the pathway have been overexpressed in E. coli, and they are able to reproduce these experiments. So if you take the lactic acid bacteria, you wash it and you mix it with linolenic acid, you will have this pathway here involving several enzymes, hydratase, dehydrogenase, mesomerase. That probably is not huge modification, but you see that you have a isomerization of the double bond here. Here this is linolenic acid. Here this is conjugated linolenic acid. So the double bond here moves from that position to that one, and you have conjugation between the two here. And these enzymes, finally, they are not so specific. They can have also this configuration here where you have two trans double bond. And it's possible that there are other possibilities. You see that in the pathway you have also this fatty acids hydroxide groups. And this is involved in the overall pathway that leads to saturated fatty acids from polyunsaturated fatty acids. So it seems to be a general way of detoxification. The fatty acids, the free fatty acids, usually they are toxic. That's why in the body they are never found free in the blood. They can be transported by albumin, in the gut you have the bile salts. So that's very rare you find at high level free, and if you find them free, you have to be careful. So in pancreatitis, for instance, we know we have a high level of free fatty acids in the colon, and this could be a reason for some inflammation. So it seems that the lactic acid buffer, they are involved in this process of detoxification. And because saturated fatty acids are less toxic, and in their way, in this function, they can generate this type of fatty acid. So this is one example. Apparently, and I am not an expert, in microorganisms, you have many enzymes for converting the fatty acids, making longer chain, elongates, making desaturation, or removing the saturation. You have also acid transferase for transferring these fatty acids to various backbones. So I think that's the next track. And to conclude, I have this slide with summarizing what we have to consider when we think about fatty acids. So it's a source of energy, and then there's beta oxidation in muscles and heart. You can store this energy instead of using it immediately, and this will be keeping it in the tissue. They can be precursor for pro and anti-inflammatory molecules. They can be transferred into, so instead of being used or stored in living bodies, they can be transferred directly in membrane lipids. So usually living bodies are also storage for the structure of cells. Gene regulation, so I did not enter too much in this, but there is a huge literature on this right now. And they can be a substrate for the GI tract microflora. And depending on the product that are generated here, you can imagine the connection with all this pathway. So it seems that there is a large interest in the synthesis or use of fatty acids in anaerobic bacteria. And this interest is not only for understanding health problems. So there is a large interest for biotechnology and using these enzyme systems for producing specifically fatty acids. So if you identify the genes and you transfer them to the host where you can produce them at high level, you can produce fatty acid at high level for industrial production. And that's all. Thank you.
Très bien. Donc, il y a plusieurs points que je voulais voir au fil de cette occasion. En premier, c'est on réfléchissait, et je pense qu'il faut avancer sur la création d'une banque, si elle n'existe pas, puisqu'il y a l'équivalence de la banque de Bernard Henri sur les cas, sur les pathways de lipides. Je pense que c'est un vrai point très important pour nous, essentiel pour ensuite les travaux qu'on veut faire sur le microbiote de l'obésité. Donc ça, on avait réfléchi, il faut réfléchir à trouver un candidat mixte comportable que pour routier ou dépose un projet ensemble. Enfin, il faut commencer ça, et donc il faudrait vraiment qu'on se mette avec Bernard Latour à essayer de comprendre qui fait quoi, de quoi on a besoin, bien humain, de quoi c'est un vrai point. La deuxième chose, c'est qu'on a une bactérie comme ça qu'on va travailler avec un liste qui s'appelle la tourette et qui donne ça, qui donne des gros poids. Donc c'est vraiment très intéressant. En parole, on fait ça. C'est facile à doser, ça, le CLA?
Oui, justement par rapport aux différentes voies pour lesquelles on va s'intéresser en priorité. Et ça, c'est grâce à ce qui est le plus simple. Alors, tomato-variété gaz, même des fois sans masse, on va identifier la plupart de ces restaurants.
Ça, à mon sens, on peut regarder très vite sur. On a, avec Maurice, on avait même déposé un brevet, je n'ai pas gardé dessus, parce que l'on développé, mais on a déposé un brevet sur le foie gras des canards. C'est-à-dire que tu as les canards en 6 semaines, qui ont le foie 2 fois plus gros, quand tu leur as donné 2 fois de ce qui s'ensuit, les doses faibles de la cholécyste figurine. Vraiment, c'est spectaculaire. Et on retrouvait effectivement un foie plus gros chez les canards, chez les souris.
Est-ce que vous avez déjà ces pistes-là sur le CLA?
On était, si tu veux, on était comment te dire, j'étais moins dans une phase explicative que dans une phase de confirmation d'une découverte par hasard qui était ce truc qui s'est grossé de manière incroyable. Donc on était plus à solidifier ça et à essayer de voir si on pouvait reproduire ce que les gens font chez les animaux de ferme, c'est-à-dire qu'ils font grossir avec la production. On était plus dans ce mécanisme-là. On avait un peu travaillé avec un intestinal ici qui trouvait qu'il y avait un profil soit d'hyperaffinité, etc. Sans aller trop loin, mais c'était à chaque fois c'était une découverte. Donc quand les poulets grandissaient beaucoup, il y avait un gros foie aussi. Et quand on est arrivé au canard, par exemple, les canards n'ont pas grandi, tu vois, mais ils avaient un foie qui était le double du foie normal. C'est pour ça qu'on avait pensé, on voulait faire avec l'INRA pour foie gras. On peut faire du foie gras tout seul sans qu'on les garde. Les amis et les amis.
On fait foie gras, on fait du foie gras avec du canard et doigts, jamais avec du poulet.
Je sais. Et donc c'était assez intéressant. Et donc ça peut amener à réfléchir sur voir si effectivement cette bactérie a un profil où on serait très intéressé pour voir si c'est très complexe.